to go.
Hi there, I'm Dan Lapworth from the British Geological Survey, and we're presenting two posters at the IAH 2014 Congress in Marrakesh. Just showing a little bit of evidence from some research we've been carrying out in Zambia as part of the UPGro catalyst grants. This is a research project that's focused in a growing urban town in sub-Saharan Africa called Kabwe, with a historic a history of mining activity and also a heavy dependence on groundwater, and also a self-supply from shallow groundwaters, particularly in slum areas. And that was the focus of our research. We undertook a water quality survey and sanitary risk assessment survey within the slum areas of Kabwe to look at the risk factors associated with the use of these shallow supplies, and to look at the seasonal variation in water quality, and to try and understand the sources of contamination in the shallow groundwater. Now, as part of that, we did some fairly novel characterisation of water quality, both looking at broad range organic contamination, and also a poster over here highlighting the potential use of a fluorescence in situ probe for predicting fecal indicator bacteria, and I'll go on to that later. To start with, so looking at emerging organic contaminants, these are contaminants such as pesticides, antibiotics, personal care products, things that are found in wastewaters. We did a survey focused on the shallow groundwater system to look at the occurrence of these types of organics. And as far as we understand, this is probably the first such study to be undertaken in Africa. So if I draw your attention to this figure to start with, figure 4, this shows the number of detections of different types of organic contaminants, both in high-income areas, low-income areas, and also in an industrial and a peri-urban setting. And the point to take home really is the high number of organic contaminants seems to be associated with these low-income areas, and we see relatively little contamination in the higher income and the peri-urban areas. So we see preferential contamination of a range of organic contaminants within this shallow groundwater system, particularly problematic in low-income areas. And this is likely to do with the density of wastewater sources in these areas, and how wastewater is dealt with, and the diffuse nature of wastewater contamination to the shallow groundwater system. If I draw your attention to this figure next, this details the types of organic compounds that we've been finding in these shallow groundwater systems. And the first point to note is that we've found this compound called DEET, which is an insecticide that's applied to the skin. We found that almost to be universally present in each of the areas we visited: the peri-urban, high-income areas, low-income areas, and also in an industrial area. So this seems to be quite a widespread contaminant in the shallow groundwater system. Second point to take home are the types of compounds that we're finding. Over here we can see a number of chlorinated trihalomethanes that are being generated in the low-income shallow groundwater system. And these are often associated with the on-site treatment, chlorination of shallow wells, where they introduce, for instance, chlorine tablets directly into the wells. If the wells have a high organic loading, then these can actually generate trihalomethanes, which are potentially carcinogenic compounds. So this is something to be quite aware of in areas where you have high organic contamination. Secondly, there are some interesting different compounds that we've seen, for instance caffeine and triptophan, which are classic wastewater indicators. These are contaminants that are associated with wastewater sources in the environment. For instance, triptophan are used quite widely in creams and also in hand washes for cleaning and for antibacterial purposes. And these are often quite resilient within the environment. They're not broken down easily because they're antimicrobial. And they're quite good traces for wastewater contamination. So it's interesting that we're seeing some of these typical compounds. Also up here, in in the higher income areas, we've also seen evidence of some pesticide contamination. This shows us that while the numbers of compounds are quite low and the concentrations are quite low, there is some evidence of urban agricultural contamination within this type of setting. If I draw your attention over to this plot here, this shows the distribution of this compound, this insecticide called DEET, within the different sampling areas, the low income area, high income area, and peri-urban. And again, this illustrates the point I made earlier, that this compound is quite widespread in the environment, and also we see almost universally an increase in loading, increase in contamination in the shallow groundwater system following the onset of the rainy season. So we see a sort of a consistent increase in concentration within the groundwater, and we think this is probably best explained by a continued source within the unsaturated zone that, as the water table rises, is being picked up in the shallow groundwater system, and the concentrations being increased in that way. This shows that the system is fairly responsive, recharge is happening very actively, and there is a rapid pathway for recharge and contamination from surface settings to the shallow phreatic aquifer. If I now move on, perhaps if I just summarise the findings. So this again, just to reiterate the point, we think this is probably one of the first studies to quantify a range of organic emerging contaminants in groundwater. DEET, this insecticide, was found universally within the shallow groundwater system, and this has also been found to be the case in other studies undertaken in tropical regions such as Singapore and Southeast Asia. Many of these compounds are frequently identified in the developed world. Were not found in such high concentrations within this particular setting, and we think this is probably due partly to the availability and also the cost of these types of compounds and the use of these types of compounds within the developing world, and also the fact that the urban setting that we were doing this research in is fairly low density compared to other areas, so may have a lower contaminant source loading compared to areas like Osaka or Lagos with a much more dense urban population. Right, so this poster highlights the use of a field probe that can quantify a protein marker called tryptophan, which has a strong association with wastewater sources. So it's a good predictor of the contribution of wastewater contamination in the environment. We thought it might be good to do a comparison between traditional fecal indicator bacteria counting and the quantified amount of tryptophan found by this probe, and see if it was able to predict both the presence and absence of fecal indicator bacteria, and also if it was able to predict the level of contamination of fecal indicator bacteria. Perhaps so it could be used as a rapid screening tool as part of water quality surveys in these types of settings. It doesn't require many reagents, it's very simple to operate, it's relatively cheap, relatively cost effective, so it might be a good tool to work alongside traditional fecal indicator bacteria counting. So this figure here just shows some preliminary results, both the relationship between the tryptophan concentration and the depth to groundwater. So we can see, and this nicely illustrates the point that the shallow groundwater systems, which have high fecal indicator bacteria counts, also have very high tryptophan concentrations compared to the deeper boreholes within this area. If we compare the tryptophan concentration with the sanitary risk score that we undertook as part of the survey work, again we can see it's quite good in predicting the high and very high sanitary risk score sites, and again we find this nice trend of low tryptophan in the low to moderate sanitary risk score sites. This figure shows a borehole profile of tryptophan down an open open screened borehole that we came across in our field area. We lowered the tryptophan probe and did a profile. This is prior to them actually installing a pump in the site. And it just gives you an indication of the vertical variation in tryptophan within the saturated zone and how this rapidly deteriorates with depth. Another technique that we thought we would pilot within this case study was the use of qPCR. This is genome sequencing of fecal pathogens. There have been very few studies that have undertaken pathogen sequencing techniques in sub-Saharan Africa, and these give, rather than generic values for fecal indicator bacteria, these give particular species level data. So as part of this study we were able to isolate the organic material in the field. We were then able to preserve this material on filters in the field and freeze them prior to transporting them back to the UK. This was found to be very robust. We carried out blanks in the fields and these were found to be robust as well, so we're quite happy with the method. And also we were able to collect enough material even from relatively low contaminated deep borehole sites. We were able to collect enough material within a realistic time frame in order to quantify the absence or presence of these types of pathogens, which is also quite useful. So once we'd taken them back to the UK and we carried out the sequencing work, we looked for a suite of 26 different pathogens, and this just gives you some preliminary results from the Kabwe case study. So this is split into the data set split between boreholes and these shallow groundwater sites, and this is a number of different pathogens that we found. So we can see again, as in the fecal indicator bacteria and also in the organic contamination, we can see a much higher level of contamination from a range of different species of pathogenic bacteria or potentially pathogenic bacteria within the shallow groundwater system. And probably to draw your attention to one particular result, this area of Zambia is quite well known for intermittent outbreaks of cholera, particularly during the wet season, and you can see that three of the shallow groundwater sites that we undertook actually had the presence of the cholera virus, as well as a range of other potentially pathogenic viruses. These two results up here for Astromyces and C. frudi are associated. These are soil-associated bacteria. So this again provides strong evidence that there is a rapid route, potentially a rapid route of transport from the soil, from the shallow zone, into these wells and into these boreholes that we've been sampling. So I think that's probably it. Probably whittled on long enough. Thank you for your time, and I hope that was interesting. We've published this research in Water Research as a sort of a full paper, so if you're interested, there's a lot more detail on that in a journal publication. Look in Water Research for that.